All right, guys. Unfortunately, I didn't bring my Surface home, so I'm going to try my best to record on my laptop at home, but I can't use my pen, so you'll just have to listen and I'll try to type some information. This paper is talking about oncolytic viruses. So these are viruses that kill cancer cells. And it's pretty amazing, I think, that these viruses can selectively kill cancer cells. So they either selectively infect or selectively replicate. And this is showing functioning cancer cells. Then they kill the cancer cells by lysis. So cancer, the way they are selective is they take advantage of genes that are expressed differently in cancer versus normal cells. And I will work on posting some specific examples today in this lecture how our virus works. So since they're replication competent, they can go back and infect more and more. These 3 slides are just tables from the review I posted. I just wanted you to understand that there are multiple different viruses researchers are trying to develop for or to become oncolytic or cancer destroying viruses. A is a vaccinia virus. So it's a double-stranded DNA virus. It's a pretty big virus. You can see an advantage is that it can hold a lot of information. So this is transgene capacity. And you can get a pretty high titer, so you can make a lot of virus for using as a chemotherapy. They're also looking at different RNA viruses. The problem with a lot of these is they don't have very big capacity because they're usually smaller viruses. Titers. And this, I know it's really hard to see, so take a look when you get a chance in the review. This is just showing you different viruses that are actually in clinical trials as cancer therapies. Looking at today, I know you can't see this very well, but it's Pexavac. And you can see that it's being used to treat melanoma, liver cancer, excuse me, colorectal cancer, breast cancer, and hepatocellular carcinoma. So let's talk a little bit about, this is the big official name of the Pexavac virus. It's based on vaccinia, so there's vaccinia, it's a pox virus. It expresses, oops, oh, this isn't going to work. Okay, I'm going to have to do it here, and hopefully this works. Is GMCSF, which I'm just going to try to write this CS. Ugh, now you're going to see my horrible typing. CSF, GMCSF, granulocyte macrophage colony stimulating factor. So what you really need to know is this is important to stimulate macrophage activity and maturation. So it recruits macrophages and it gets them going. Remember, macrophages are those types of cells of our immune system that can phagocytize, so it can engulf large chunks and get rid of foreign objects in our body. This virus also has what it shows you here, and most of the time you just see this written as TK. So thymidine kinase, a kinase is something that a protein, an enzyme that phosphorylates. So thymidine kinase's activity is to phosphorylate certain molecules. And its highest activity is in the G1 to S phase, which means thymidine kinase is a cell cycle marker, which means that if you have an increase in thymidine kinase activity, you have an increase in cell division or cell proliferation. So in cancer cells, you see thymidine kinase upregulated or expressing at higher levels because the cells are going through cell cycle very quickly. And so the way this virus is specific to cancer cells is that it is only going to replicate in cells that are expressing high levels of thymidine kinase, which would be cancer cells, not normal healthy cells. So that's how it's selective. You're going to see an NP before the JX594. This, oops, wow, 594. This just means that it's a mouse version of Pecavax. Or Pecavac. I don't know why I want to call it Pecavac. So the idea of this oncolytic virus is to not only kill tumor cells or cancer cells, but also using GM-CSF, immune cells to the tumor to help continue destroying it. So just a teeny bit about cancer. Cancer is basically uncontrolled cell division. So you mass tumor can be invasive, which means it can push on other tissues and cause issues. Once the tumor gets above, is pretty dang small if you look at a ruler, it actually needs its own blood supply. Oops. So this is called angiogenesis, and that is making your own blood supply. And so tumor cells, cancer cells, will secrete factors like VEGF, which we'll talk about in a minute, which will start promoting formation of this blood vessel. And then once these tumor cells are fed through the blood vessels, they can grow bigger and bigger. So one of the things that's the challenge with cancer is you have to cut off that blood supply to the cancer cells. So invasion is just staying in that area but kind of pushing on neighboring tissue. Metastasis is where you actually get a cell from the tumor or the cancer cells to detach and move. Cells break free into the bloodstream and start growing a second tumor. So metastasis, those are the scary kinds of cancers because they can move throughout the body, and so then they're really hard to control because we don't know where to go. A little bit more about the vasculature system. One, it's kind of interesting, it's very disorganized compared to normal tissue. But again, I mentioned this VEGF, which is vascular endothelial, oh, that will just go right off the screen, growth factor. And this promotes formation of blood vessels. So this is something that is upregulated in cancer cells. Sunitinib is a chemical that is an inhibitor of angiogenesis. So inhibitor of making that blood supply. Sunitinib works by blocking the VEGF receptor. So one kind of interesting thing is whenever you see NIB in a cancer drug, NIB means it's a it blocks receptor tyrosine kinases. So hopefully you learned about these receptors in cell biology. So there's lots of different drugs with the ending NIB, and it means they're a receptor tyrosine kinase blocker. Sunitinib is specific to the vascular endothelial growth factor. So it can block the activity of VEGF and block or inhibit angiogenesis, or the blood vessel growing. And the other thing you'll see in this paper that relates to VEGF is you'll see this DC-101. This is towards the end. This is an antibody that blocks the VEGF receptor. So this antibody, we talked about this in our last paper about antibodies blocking the receptor so that you can't get the signaling. So they're testing the importance of VEGF that blocks that pathway. Okay. The two type of mice that they're using in this paper, the main one is the RIP tag, and RIP stands for rat insulin promoter. Okay, so that's the promoter for the gene, and the gene that it's controlling is the large T antigen from the SV40 virus. So TAG stands for large T antigen, is what we call an oncoprotein. So it's going to be cancer causing. It promotes cell division or cell proliferation. So this is, again, characteristic of cancer is that the cells proliferate or divide uncontrollably. So this RIP tag means that the oncoprotein is going to be expressed where insulin is normally expressed. So that means that in the pancreas, the beta cells are the insulin producing cells. Okay, pancreas. I don't know why I always put it either. Or the insulin producing cells. So this is where our cancer is, in the beta cells of the pancreas, and I'll show you that in just a minute. These tumors show lots of angiogenesis. So what's kind of interesting is cancer of the beta cells accounts for only about 5% of pancreatic cancer. So they're not really using this as a model for pancreatic cancer, but they're using this as a model for angiogenesis and trying to test what types of viruses or cytokines or different drugs will stop that angiogenesis. So the type of cancer that's in here is called insulinomas. So cancer of the insulin producing cells. This other guy over here, he's called a nude mouse. And he has no thymus. So that means he does not make T cells. And so a nude mouse has a very inhibited immune system. And they don't show data in our paper from this mouse, but this is all in the supplemental. And they do something called a xenograft, which is where they are putting cancer cells into these mice and then looking at treatment. And a lot of times they're putting human cancer cells into the mouse. So xeno means foreign, graft means like putting it into, getting it to grow in the organism. So a lot of times they'll put human cancer cells into these nude mice and then test for a drug. Anyways, it's in the supplemental, but most likely we're going to get to some of the supplemental figures next week. So here's the pancreas, and here's the small intestine. So the pancreas's function is to help convert food to energy. So there's two types of cells. The endocrine cells, which are our, found in the islets of Langerhans. They make insulin and glucagon to regulate blood sugar levels. Okay, so this is actually only about 5% of the total pancreas is the endocrine cells. Most of the pancreas are called exocrine cells. These are the acinar cells, and these are actually making digestive enzymes. So the one that they will talk about in the paper is amylase. So they break down carbohydrates, they produce stuff that breaks down lipids, breaks down proteins. So it helps our digestion. So let's look at those cells. Okay, so here is your endocrine producing cells. Interested in is the beta cells, so you're going to see staining for insulin. And remember, this is our cancer. The acinar cells are all these yellow ones around. It's a lot more of the tissue. They're producing amylase as one of the proteins, and you'll see that so they really surround our cancer cells in there. You're going to see this kind of staining in figure 2A, 6F, and 7A. Okay. So they are going to look for different markers that signify tumor destruction or signify that we're having a positive effect on the cancer. So one, they're going to look at leaky vasculature, and they're going to use nanoparticles. They also talk about fibrin and red blood cells. Okay. And basically what this is trying to show you is that a normal vasculature lining, the endothelial lining is nice and tight, and so not many of these nanoparticles are going to get through. But when you destroy make them leaky, and so you're going to see an increase in nanoparticles in the tumor tissue. And extravasation is basically the word that talks about leaky. So things are coming out of the vasculature into the tissue. Vascular pruning, just think about like if someone's pruning a bush, they're cutting it back. So I like this picture because this shows you normal vasculature. This shows you vasculature in a tumor. And so you can see it's not as neat and organized. And then what they're doing here is they're using anti-VEGFR, which we talked about. VEGF is a promoter. They're blocking the signaling of the receptor to block more endothelial, more vessel production. Adequate vasculature system for tumor growth. So remember, once it's bigger than 5 millimeters, it needs additional blood vessels in there too. Well, be looking at apoptosis, and they measure the caspase activity. So this is just showing you that there's a whole bunch of different caspase proteins that are involved in promoting apoptosis. So they'll measure apoptosis by staining for caspase. And remember, apoptosis is programmed cell death. So you're trying to get rid of these. Reminder about some of the cells of the immune system. If you see the term leukocyte, that just means all your white blood cells. So that includes these cells of the immune system, the B cells, and the T cells. They talk more specifically about T cells and these markers. I'm just going to write a couple things. So all white blood cells are CD45 positive. Okay, so monocyte, B lymphocyte, just like we saw, all of these are going to be CD4 positive. When they stain for that, or when they count that, that's just counting all the white blood cells that are in that area. When they look at CD3 positive, those are all your T cells. Okay, so you can see your helper T cells, your cytotoxic, your activated, those all have the CD3 marker. When they talk about CD4 positive cells, those are your T helper cells. And so those activate macrophages and increase inflammation. Then they're going to talk about CD8 positive cells. Those are your cytotoxic. See, it's over here. Cytotoxic T cells. Those destroy other cells. But they can also increase inflammation, and they by releasing cytokines. And then the last kind is over here, CD25 positive cells. Those are called the Tregs, the regulated T cells. And these are important for preventing autoimmune diseases, like recognizing self. The problem is what they've seen is that they are upregulated and recruited to tumors. And so if you have a high level of the CD25 positive cells, it's usually an indication or of what they call poor prognosis, which means your body is, these Treg cells are telling the other cells, no, this is okay, this is self, don't destroy it. So it kind of fights against your body's ability to prevent the tumor growth. The other interesting thing about Tregs is they produce something called granzyme B. So let's look at that. Granzyme B is called a serine protease. So it cleaves proteins at the serines, and its job is to activate apoptosis. So I'll be honest with you, I think it's a little confusing about the role of Tregs or Tregs in cancer. In one hand, it activates apoptosis, and so that would increase tumor killing, but on the other hand, they say that if you have a lot of Tregs there, and I think they're called Tregs, but I'm not an immunologist, so I'll probably call it wrong, they might stop your body from attacking the tumor. Anyways, granzyme B is here in the yellow, and it activates these caspase enzymes, apoptosis. Tumor burden is another way they measure. So is the tumor size decreasing? And what I want you to remember, this is from figure 2A, the red is the insulin producing cells. These are our cancer cells. And the amylase in green is the surrounding acinar cells. And so you can see here that as you're decreasing the amount of cancerous cells, and it's shown here in the graph. So just kind of keep those in mind as to why they're staying for insulin and amylase. Insulin's our tumor cells. Amylase is the normal tissue around it. They do a lot of histochemistry. So all of this, the red and the green and the boxes, they're using antibodies to specific proteins that have red or green fluorescent tags, and that's how we can visualize. So this just lists some of the ones to remember. I saw that Sarah started kind of a abbreviation page, which is a great idea. So you guys could add these to it. So anytime you see CD31, they're looking at vascular tissue. Is our oncolytic apoptosis, and so on. What I also want you to understand is in immunohistochemistry, when you're using red and green, you will also see yellow, and that just means that's where they overlap. So yellow is not a different thing. It's just where both red and green are. So this means wherever there's yellow, there's a lot of apoptotic cells and the vascular marker for CD31. They also use flow cytometry. Just to remind you, when you look at these flow cytometry figures, things are broken into quadrants. Okay? So it will tell you the markers on the X and Y axis, and the top right quadrant are cells that are positive for both markers. The bottom left are negative for both markers. And then these other quadrants, like here, these are CD19 positive, but CD34 negative. And these are just the opposite, CD34 positive, CD19 negative. So this is always how the quadrants are labeled. Here's an example. I forgot to put what figure it's from, but this is from our paper. And so again, CD45, always forget. CD45 is for the white blood cells. CD3 is telling you what population of those white blood cells are T cells. Here are the white blood cells that are T cells, right? Double positive. And it makes sense. You can't have a T cell that's not CD45 positive. These are all the cells that are not white blood cells because they're CD45 negative. And these are all the white blood cells that are not T cells, so the B cells and the granulocytes and the eosinophils. So keep that in mind. You're always going to see these quadrants, and you're going to see numbers that are talking about the percent of total cells in these quadrants. The intensity, so blue is the lowest intensity, so it's just showing you there's a lot in there. Instead of it being just a solid blue blob like here, these are actually showing you kind of levels. Supplemental figures in this paper, and we'll tackle the figures that we see right now first, but as you're reading the paper, if you find interesting supplemental figures, make a note of it, and we will focus on the supplemental figures on the second week. All right, thank you, and I hope this worked.